All right, there we go. Okay, so tonight my goal is to help demystify leafy liverworts with hints about how to tell some genera apart that superficially appear to be very similar. To do this for all leafy liverwort genera would be way too long a topic for one bryophyte beginning session. So I thought I'd focus on those genera in our area, meaning our area, the southwest to California, that have bifid or two-lobed leaves. I've struggled with many of these, and based on the determinations I've seen in herbaria, a lot of others have too. In Colorado, such liverworts generally grow in wet areas in montane or subalpine forests, hence the title of the talk. So, just a little review. Unlike the mosses, which all have rather similar leaves, leafy liverworts vary dramatically in the size and the shape and the arrangement of leaves. Most, but not all, leafy liverworts are somewhat dorsiventrally flattened, and in dorsal view, we usually see two ranks of leaves. And if we look on the ventral side, such as here, this is a species of Bassania. We see the lateral leaves, and then on the underside, there's a third row of leaves, which are not at all like the lateral leaves. They can be in some liverworts. All three rows of leaves look the same as in Trichocolia down here, an eastern liverwort, and then some have no leaves at all. The way the leaves overlap is a really important character to learn. If they're succubus, they're arranged like shingles on a roof. And if they're, and you can see the bottom edge of the leaf, if they're incubus, then it's exactly the opposite. It's easy to know as long as you are sure which is the apical end of the stem, because if you get the wrong end, you'll get them completely backwards. But as long as you know where the apex is, those are pretty easy to tell. As in the mosses, the leaves in leafy liverworts are source of many, if not most, of the characters that we use to tell one from another. Of the 33 genera of leafy liverworts that grow in the region from the Rockies to the West Coast up to the Northern California border, most, that's 18 out of 54, I mean 18 out of 33 or 54% have bifid leaves. So there's a better than 50-50 chance that a leafy liverwort you encounter in the field has a leaf with two lobes. So if you can learn to tell these apart, you have a leg up in coming to an accurate determination, at least to genus. We'll get the easy ones out of the way first. Some leafy liverworts have bifid leaves that are what we call complicately bilobed, which really just means that the lobes are unequal. One lobe is much smaller than the other, and the smaller lobe is pressed up against the larger one. In the genus Scapania, shown here, the smaller lobe is on the dorsal side. It can be sharply folded or kind of lax. These plants are rather lax in their folding. Um, in the others, in Radula perella and Frullania, the smaller lobe is on the underside. And so you can't see that smaller lobe unless you remove the liverwort from the substrate and turn it over. In Radula, that smaller lobe, it's rectangular or triangular, and it's fused on one side to the stem, forming a little pocket. And Radula has no underleaves, so you can see it quite clearly. In Perella, the lobe is it's not attached to the stem except it's just attached to the lobe, the lobule or the smaller lobe is attached to the lobe base, and it's kind of ligulate in structure. And it can be hard to see because Perla has very large underleaves. So to really see the ligulate lobules, it's a good idea often to remove some of the underleaves, but you can see them here. And in Frullania, it's the most complicated system of all because the lobule has become modified into an upside down sort of a cup-like structure. It can be kind of like an upside down urn, or there can be broader ones that are more like a helmet. Um, and Frullania has bifid underleaves. Again, to see the attachment of the lobule, it's often a good idea to remove some of those underleaves. So these groups are all very easy to tell apart at the genus level, but species determination is quite difficult. You can see they are all quite large, not so large in our area, but still kind of challenging to determine the species level. I want to say before going on that a lot of the a lot of the good images in this presentation are from David Wagner's Oregon Liverworts publication. I credit it just by saying Wagner to save space on the on the screen, but he takes really great pictures that I am taking full advantage of in this talk. So most of the genera of leafy liverworts that have a simple bilobed leaves, most of them have simple bilobed leaves, which means that the lobe is in one plane. The lobes are more or less equal in size and shape. The features that you need to pay attention to when looking at these bifid leaves is how bifid are they? You see, they range from being emarginate to shallowly bifid, as in this leaf, or very deeply bifid, as in this one. Many of them are somewhere in between a third and a halfway bifid. And other important characters are the orientation. The lobes can be parallel. They can be divergent if you were to draw a line from the base of the leaf to the through the pointy part. Um, and lobe shape, whether they are rounded or attenuate, as in this one, and the lobe, the sinus shape, whether it's U-shaped or V-shaped. So those are the kinds of characters that that we can get from bifid-leaved liverworts. We'll start with the pair of Gymnometrium and Anthelia, both of which grow in the West, usually at high altitudes and latitudes, or yeah, high altitudes or latitudes. I've not collected Gymnometrium in Colorado, but I come across Anthelia fairly often on exposed soil areas, often above tree line, 12,000 feet or higher. These two genera, like many of the groups of genera with similar leaf lobing that I'm going to show you, are not considered to be closely related at all. In fact, they belong to different families. And the familial classification in liverworts has traditionally been based on sexual characters, much of which was kind of confirmed by DNA analysis. But so not leaf type. That's kind of a convergence. Both Gymnometrium and Anthelia are very small and they're sort of wormy looking. Anthelia plants often have a appear to have a whitish coating on the surface, as you see here. It's actually a wax that's exuded from the leaf cells, and it probably helps the plant deal with this intense radiation that it gets at the high elevation where it grows. Gymnometrium doesn't have a waxy coating, but because again it grows in these highly exposed areas, the plants often look kind of bleached. And if you did not know what the white coating in Anthelia looked like, you could confuse the bleaching in Gymnometrium with that. Um, with microscopic examination, the two genera can be distinguished pretty easily. Anthelia is one of those genera in which the underleaves look identical to the lateral leaves. So it has the same leaves going around the whole stem, whereas Gymnometrium has just two rows of leaves and no underleaves at all. Um, the leaves in Anthelia are deeply bifid, almost to the bottom, whereas in I'm sorry. Um, whereas in Gymnometrium, come on, go back. Oh. Whereas in Gymnometrium, they're only sort of shallowly lobed. And this, honestly, the leaves are different enough here that really once you see them, you think, why would you confuse them? It only comes because the plants are so tiny and they are hard to work with. And you might miss the differences of lobe depth if you were just looking at them without a good microscopic presentation. Okay. Cephalozia and Cephalosiella are two tiny bilobed leafy liverworts that can look similar, and they have similar names, but they aren't considered to be related at all. They're in different families. They do often grow in similar habitats. I collect them in Colorado, often in wet areas, subalpine forests, again, near streams or other continual sources of water. Most species of Cephalozia are larger overall, and the lobe shapes range from incurved or sort of semicircular with a rounded sinus to plants where the leaves are only just a few cells wide. Cephalosiella almost always has leaves that are uh, have divergent lobes. They can be ornamented, as in Cephalozia divaricata, that has some little teeth on the margins, or they can be smooth. But you can see, if you look at Cephalosiella and Cephalosiella hampiana and Cephalozia montania, you can see that they look very, very similar. And it could be hard to tell them apart, and just based on dissecting scope or just even cursory microscopic compound microscope examination. But if you're working with living plants, if you've got younger or plants that are freshly collected, you can tell them apart very easily because Cephalozia is one of the few genera in our area, maybe the only one, well, at least only one of the few, that has no oil bodies at all. Here are the leaves. You can see lots of chloroplasts, but there is not a single oil body to be found in the leaf of in a leaf cell of Cephalozia. Whereas in Cephalosiella, they're tiny, but there's all kinds of oil bodies. So that's a very surefire way to tell them apart. If you have dried material, another way to do it, not an easy way, I admit, is to take a stem cross section. Hard to do in these tiny plants. But Cephalozia has a hyalodermis, which honestly you can kind of see here. You can kind of get the sense that the outer row of stem cells is rather large, much larger than the inner ones. In Cephalosiella, you don't see that. I recommend being sure and making a leaf cell, stem cross section, but that will tell you for sure. There are a few regional genera, each represented by one or just a few species that have succubus flattened leaves that are bifid to about 1/3. And these include Lophicolia minor, which is widespread across the United States, as is, oops, sorry. Um, as is Geocalyx graveolens, again, widespread across the country. Harpanthus has three species, two of which, Phlotobianus and Drummondii, have been collected in the West, but not very commonly. And then Mesoptychia, the genus, Mesoptychia bantriensis is, well, the genus Mesoptychia overall has 4 species that occur in the West, one of which is endemic to California. It sometimes goes under the name Lyocolia or Lophosia in the older literature. Um, Now, from a dorsal view, they all look pretty similar, so you got to turn them over and look underneath, except that Lophosia minor, at least in my experience in Colorado, almost always has gemmy on the leaf margins, bright green gemmy on the leaf margins. So that's a pretty good way to tell that. If you come across one without gemmy, if you look at the underleaves, in Lophicolia, the leaves are bifid. The underleaves are bifid. They're kind of, the lobes are very skinny, and it's got a little auxiliary tooth on either side of the underleaf. Geocalyx graveolens doesn't have gemmy. It does have bifid underleaves, as shown here, but they're larger. They're more substantial underleaves and without any sort of a side tooth. In both cases, in both Geocalyx and in Lophicolia, the rhizoids are restricted to the bases of the underleaves. In Mesoptychia, the underleaves are pretty tiny usually. Here's an underleaf. It's just a few cells, although they can be bigger than this. Another way to tell it is that when you mount it under the microscope, here are the cells. You see those little mottled markings. Those are strong, they call it verruculostriate. They're irregular cell wall thickenings that you can see readily at 40x magnification. And that's, they're just jumps out at you. So that tells that one. Harpanthus has underleaves that are large compared, say, to Mesoptychia, and they're often attached to one of the leaves. They're connate with one of the leaves on one side. It's hard to show this. I think we can see right there it's actually attached, but I find this a very hard character to demonstrate because it's very hard to remove both and not pull them apart. Harpanthus underleaves are entire. They have some little side branches. I have had a hard time with this genus and confusing it with Mesoptychia because even though what I've shown in Mesoptychia here is a very small underleaf, they can be larger and they can have little side branches, and both can have a verruculose striate margin. Now, if they're fertile, it'd be super simple. Mesoptychia has a big perianth that is has no keels at all. It's kind of like a balloon. And Harpanthus doesn't have much of a perianth. It has more of a sort of embedded in the plant, kind of with a fleshy base around it. But I have struggled with these. Um, okay, excellent. Classically, the genus Lophosia was the home for species of liverworts with succubus bifid leaves, with no underleaves or very tiny ones, and gemmy at the leaf apices, as you can see here in this one and here. Those are the gemmy. Lophosia is generally considered taxonomically to be one of the most problematic genera of leafy liverworts in the Northern Hemisphere, with more than 60 species. Phylogenetic studies based on chloroplast genes have shown that Lophosia in the broad sense is not a single unit, but and it's been divided up into a number of different genera, four or more, depending on how far back you want to go. There are 8 species reported for the segregates Lophosia, for Lophosia and its segregates Lophosiopsis, Isopachys, and Obtusifolium in Colorado and all of these, if you were to go to flora of North America, would be treated as Lophosia because honestly, it's not very easy to describe, to differentiate these genera based strictly on morphology. But Lophosioid liverworts often form tiny dense turfs in wet areas. I find them frequently in wet subalpine forests over soil or over dead wood or near streams or in low wet areas that are somewhat protected in the alpine zone. The plants generally have shallowly bifid leaves with parallel or somewhat divergent lobes. The types of characters that are used to tell them apart, the genus level, are sexuality. For example, Isopachys is generally paroicous, whereas the others are all dioicous. You got to be lucky to be able to determine that. Gemmy color, Lophosiopsis, and a few others have red gemmy, whereas Lophosia in the strict sense has green gemmy. And in all of the groups, the gemmy are angular and two-celled, usually two-celled. You can find the odd one-celled one, but generally they're two-celled. Oil bodies are another source of characters if you have fresh plants. Obtusifolia has like 50 oil bodies per cell, little tiny like droplets, whereas most of the others will have 6 to 8 kind of lumpy, elongate oil bodies. The stem structure can be helpful at the generic level in some. So this is part of a stem section in I think in Lophosia ventricosa. And this is the lower side of the stem, the one that's in contact with the soil where the rhizoids are. And in the true Lophosias, there's often a zone right next to the soil that's discolored. It's not clear. It's brown or kind of purplish. And the reason for that is that these cells are full of mycorrhizal hyphae. So there's fungi in there, and I didn't get close enough. I have blown them up when I've had a really good section to the point where I could almost convince myself I could see individual hyphae. But anyway, that is something that is characteristic of the true Lophosias. I find this group to be extremely difficult because, as I've just told you, the differentiating characters are subtle on the best of days, and the plants tend to be very variable depending on the habitat. Most of the species that occur in our area also occur in Europe and across the Arctic Alpine. They're kind of whole Arctic, I'd say. And they've been the subject of intense study for more than a century. I mean, a lot more. Schuster's treatment in the flora of Eastern North America is close to 400 pages long. This is, I don't know if you can see this. This is my focus thing. But anyway, this entire volume 2 is about 1,000 pages long, and 400 of those are devoted just to Lafon. Okay. So a few years ago, I went on a collecting trip with some of my colleagues at at the at the Denver Botanical Garden, and we went to a place in the Front Range, Colorado, to a spot which had been highly impacted by silver mining. It was really rather contaminated, although the forest has come back. And one place we collected was along the Snake River, which originates near Webster Pass, if you know the area. And this is the stream. You can see there's a reddish cast to all the rocks and this soil, this stream bottom because of the iron that's in the rocks. But this particular stream is lined with a black masses of what turns out to be two species of liverworts that are growing kind of intermixed. One is Gymnocolia inflata, and the other is Marsupella sphaculata. And I collected heaping quantities of these. And I found as I sorted them out, I thought I should know these apart. I should be able to tell these apart very easily. Since both were sterile and without a compound scope, I had a hard time. I had a hard time distinguishing sometimes. So when you look at them most closely, Marsupella sphaculata, which is the species, is generally much larger, but you can see that the side ranges do overlap. Um, Gymnocolia plants have a similar look. They're also, both are kind of brownish. They form these loose turfs. And when you pull off an individual leaf, they're both bifid. Now, this particular plant of Gymnocolia has much more pointy lobes on the leaves than is typical for the species. In fact, this may be variety acutifolia, although that's never been collected in the Rockies, but it fits that and also the fact that it's always sterile, whereas many times Gymnocolia has these enormous perianths that make it very easy to determine. But this population didn't. And the leaves, although not exactly the same, you can imagine that in a description and a key that it could be hard to use unambiguous wording that would help you be sure you have one or the other. So you have to turn to the microscope. These are the cells of Marsupella, and they have usually brownish walls that are quite thick. The walls aren't just thick, but they have what we call very large trigones, the corners where the cells meet, and they're described in this species as convex, meaning that the edges of the trigones bulge out. I have a little bit of a hard time demonstrating that here. There, if you use your imagine, you can see that they're kind of bulgy. And also, as in Mesoptychia, there's a cell with no content, but you can see on the outer, you can see those little mottled gray markings, which again is indicative of these thickenings, these irregular thickenings. In Gymnocolia, on the other hand, the cells can be rather thick-walled, but they do not have those convex trigones at the corners. The oil bodies and Marsupella also are lumpy and elongate, whereas they're more round in Gymnocolia. But again, this can be a little difficult. Stem sections will tell you for sure. I'm sorry, I apologize for the quality of this photo, but anyway, this is the stem in Marsupella. It's got quite a complicated stem anatomy. It's got an outer row of hyaline cells, and then it's got cells inside of that which have very thick walls, which are very thick walled, and in the middle there's a core of thin-walled cells. It's a massive stem. Whereas in Gymnocolia, we also have a hyalodermis or a row of larger cells. The cells to the inside are much simpler. There are many fewer of them, and we don't have that row of thick-walled cells. So you can tell them apart, although again, they shouldn't be hard to tell apart, and they certainly wouldn't if they were fertile, but I get confused by them. I have gotten confused by them. So we'll finish with the rare endemic liverwort with bilobed leaves, which has been confused with many others. This is Rivularia gemipera, which was originally described by Alexander Evans from the Uinta Mountains in 1938. He placed it in the genus Chyloscyphus because it had succubus lobes and small underleaves. But Rivularia, unlike Chyloscyphus, has dimorphic stems. It has prostrate stems, which are quite flattened and have rhizoids on the underside. This is a cross section of one of those prostrate stems. You can see it's not round at all. It's very much flattened. And then it produces that kind of stem, and then it produces upright stems, that lack rhizoids and have large to small underleaves. Here's one of the erect shoots. This one happens to have a sporophyte on it. Another feature that's diagnostic for this genus is it has fasciculate gemmi. So it has gemmi that look like a chain of beads or pearls. It can be even a branched chain, but they're all attached to one another, and then they're attached ultimately to the base of the leaf. All the other liverworts in our area have gemmi that are, they may be one or two celled, but they come apart from one another. They don't form chains. That's one thing that makes this quite distinct. It's aquatic. It grows attached to pebbles in streams. In addition to the type collection from Utah, it's been collected in Alaska, in the Aleutian Islands, once in California by me, and a number of times in Oregon by David Wagner, mostly in the Three Sisters Wilderness area, and he's the one who recognized it as a new genus. Given its distribution, I think there may be more collections. They may be hiding in herbaria under the wrong name. Wagner suggests that in addition to Chylocyphus, this plant could be confused with the Urmania or Marsipella, which is where I tried to place my collection. I wasn't very happy with it, but it was the best I could come up with. But again, the gemmi would distinguish it from any of those, since none of those other genera produce gemmi. So I hope you'll be on the lookout for Rivularia and all the other bilobed leaf liverworts. If you need help identifying them, let me know. I can point you to some resources, or you can send me pics or the specimen if you want. I can't guarantee in every case I can get you to a species determination, but I can certainly do my best. Okay, thank you very much.